They mentioned transmission, prevention, reaction, neurological complications, relapse, drug resistance, using advanced technologies. When you say mainly PCR-based technologies, when I say PCR, I'm sure for most of you it may be sounding Latin. So I would like to briefly say what is PCR and what is the, funda or the basics of PCR technology. That is, and also sequencing of Leprosy has been the major advance in this decade. So that has helped a great deal in understanding not only the bacterial genomics, its structure, and then it has applications of the same in the field and in developing other technologies for diagnosis and other prevention point of view. So what is PCR? I thought I'll just briefly tell you as to what is PCR. The idea occurred to Dr. Kenny Mullis in 1983, who received a Nobel Prize for chemistry in 1993. PCR is simply amplifying bits of DNA, and that large quantities of DNA are produced from small quantity. It amplifies in billion folds. Then analyze single DNA fragments out of large complex mixture. For example, human genome has a mixture of 12 billion 300 base pair fragments. So if you have to amplify and study each and every fragment, then you can imagine the quantum of work that goes into the whole exercise. The altered DNA sequence and side direction mutagenesis is one of the applications of it. What is PCR? The reaction components. Target DNA contains the sequence to be amplified, and you have the pair of primers, that is forward primer and the oligonucleotide that define the sequence to be amplified. You have the DNTPs, DNTP DNA building blocks, and then you have the thermostable DNA Taq polymerase enzyme that catalyzes the reaction, and then you have the magnesium ions that is coacts as a cofactor of the enzyme. And then you also have the buffer solution that maintains the pH and ionic strength of the reaction solution suitable for activity of the enzyme. These are the various steps in involving PCR reaction. They also have the denaturing at the 95 degrees centigrade, annealing of the DNA, then extension that is at 72 degrees. So this all has done mechanically. This is nothing, the technique is well standardized and well mechanically done. There is no complication about it, and it is a very simple, straightforward laboratory technique. But the main thing is that since it is a very sensitive technique, one has to apply, do it in a very ideal conditions, and any cross-contamination can give you all sorts of haywire results. So the main thing is the technological requirements. So this is what looks like a DNA, annealing DNA look like. The main DNA standard, and then you have this annealing of the DNA that takes place. So two strands made into single strand DNA, and then that is made into bits, and then it is amplified. The main applications of the PCR technology, which is actually the major advance of the current in the last decade, is mutation screening, drug discovery, classification of organisms, genotyping, molecular archaeology, molecular epidemiology, molecular ecology, bioinformatics, genomic cloning, site-directed mutagenesis, gene expression studies, and on the applied aspect of it, genetic matching, detection of pathogenesis, prenatal diagnosis, DNA fingerprinting, and gene therapy. So there are series of applications which is there is a major advances made using this PCR technology. So that is why there is a spurt of activity in this area. Now, genomic sequence of M. leprae that was completed in 2010 was the major advance in the field of leprosy, and that has helped, as I said, to explore many other areas from the point of view of early diagnosis and so on. This was completed with Stewart Cole, you all may be knowing, comparative analysis with M. tuberculosis has established that gene decay in M. leprae have resulted in 1,116 non-functional pseudogenes and elimination of many key metabolic activities of M. leprae. So that itself explains why M. leprae is so difficult to cultivate. Now, you must be all knowing, till today there is no cultivation of M. leprae. So this is one of the reasons that is being projected as the reason for non-cultivability of M. leprae, because it is almost lost half of its functional genes. M. leprae has undergone a reductive evolutionary process through adaptation to its intracellular milieu parasite. So it's only can survive as an intracellular organism. And then this is what it explains as to. It also takes 13 days to replicate. So it is a very slow dividing as compared to M. tuberculosis. So this is one of the reasons is that gene decay. Now, the advances in the field of leprosy using the PCR technologies, which are under exploration and study that are ongoing, are identification of bacterial host as well as genomic and protein markers from the bacteria as well as host for the detection of high-risk groups, difficult to diagnose cases, and subclinical infection. These are prime areas. One is looking for some age where you can have a quick diagnosis. You can diagnose subclinical infection would be of prime interest to all clinicians, all of us for that matter. One of the challenges has been to determine exposure versus infection. So how do you differentiate or decide this is only an exposure, he will not develop into, because we know that 99% of the person, individual or the human who gets exposed do not get disease. It's only hardly less than 1% develop disease. So how do you determine if you have a tool, how do you determine this is only exposure and not, this is disease versus this is exposure. So that is the main challenge in the sense in terms of laboratory thing or developing a tool or having a tool. Antigen targets that detect subclinical infection and utility of these tests in the field setting may be evaluated through the prophylactic intervention. This is one of the research areas that are being explored in the sense you develop a target and then see whether it will, because in anything in leprosy is slow progressive, including, besides its own long generation time, to determine whether the patient will develop a disease in future or something like that itself is a big question mark in the sense it takes years together for a person, between exposure and development disease, there is a lot of incubation time is from 2 years to as long as 10 years. So if that is the case, is there a test or can there be a simplified tool that can decide, okay, this man is more susceptible to develop a disease versus this man will not develop a disease, is exposed but will not. So this is the whole funda behind as to developing the tools. There are a number of host genes, for example, toll-like receptor 1, toll-like receptor 2, these are the membrane-associated receptor and clusters, LTA gene, HLA gene, interferon gamma, vitamin D receptors, this part 2 and part G, these are all being looked into in the sense because they also have shown potential in terms of determining, and some from the host side, host genomic factors have been studied for to see the difference in different population conferring susceptibility. They want host genes are being studied, whether they confer susceptibility to an individual. That's one area that has been explored in the laboratory side of it. So these are some of the genes that are coming from the host. An application potential of present knowledge on strain typing M. leprae, for example, M. leprae genome is in open now. So using certain marker SNPs, there, for example, SNP typing and VNTR, various number tandem repeats it is called. So these are useful tool that has been identified in tubercle bacillus as well. So they are trying to extrapolate it to M. leprae and see whether these variable tandem repeat areas will be useful for M. leprae strain typing. As such, till today, there are hardly 3 or 4 different types of M. leprae strains have been identified in India, and they are still in the thing, there is not too many strains, that is still not very certain whether they are really a difference in the strain, or is it just a sort of a, I mean, like the deletion that is happening, maybe there is some difference, or one has to confirm that it is really useful in strain typing. Will it be useful in transmission? Can you use this technology knowledge in studying the transmission, using particularly probably in along with geocoding system? Because if you have to identify a cluster with some different strains, or is that strain is responsible from, I mean, transmitting the disease from one area to other area to other. So you have to follow the cycle and see, and then do a grouping and how the transmission happens. For example, if you have a drug resistant strain, if you have identified DDS resistant strain, then if you have to follow whether that particular strain is getting transmitted from one individual, one cluster to another cluster, you will have to follow the cycle. So it's a long process, but it is being tried in the laboratory level, not at the There are some Leprosy specific genetic markers like 16S RNA in the and RLEP and TTC repeats in PCR based assays, that is also have the potential of use in detecting viable bacteria in a given individual. So that can have multiple uses. In the sense, if you detect, it can be used as a marker for monitoring treatment. For example, we don't have a treatment endpoints today. In the sense, when you say, when a patient is released from treatment, you give him a fixed duration treatment and say patient is released and cured. So that's the definition of cure. But it's not really a definition in the sense from the scientific point of view. I mean, one need to know, is it really, I mean, the patient is all, you know, he got rid of all the viable bacteria. That's most important from the disease point of view, infection point of view. Is all the bacteria are dead? That is the question that one need to ask. So these may be some of the markers. For example, 16S RNA is a viable marker. And using a certain, is that if you can detect and say the level of this particular Leprosy marker, can we use that as a marker for saying, okay, this man is cured and there is no viable bacteria in the system. So is that's one of the aim of this test that is being developed or is being aimed at, and one is trying to see whether it can be made it fuel friendly. Can you have a simple technique like a dot blot assay or something like that to see, okay, this man is cured, now we can release him from the treatment. So that is where probably we need a test. Now there are skin test antigens that have been tried for now almost almost 10 to 15 years now. There are a whole group of people working on this skin test antigen to detect or confirm early diagnosis. I mean, to say this patient is a case of leprosy, early diagnosis, particularly in indeterminate case of leprosy or doubtful cases or monolesional cases. Can you confirm? Is there so? There is one group which is trying to develop a skin test like Lepramin test or a Vitsura test and something like that. So skin test using Leprosy specific antigens. So they have proposed, or they have in pipeline, antigens like MLSA, LAMP and MLCWA are used as a skin test antigen, are being tested. I'm not saying that it is already in application in the field, but it is as a laboratory level has been proposed as one of the complex. There are also Leprosy specific antigens which are complexes which may have a better application being tried. Now there are serological markers like PGL1, 35 KD and 18 KD antigens have been studied. And it is shown that in some of the lab-based studies and in some of the small scale-based studies, 90 to 95% of the BLLL patients and 25 to 60% of the TTBT cases were found to be positive for PGL1 antibody. So this has good sensitivity, but we still find that it is not from the, because as you know, 70-75% of the cases are actually borderline tuberculoid or smear negative group. So this the application will be very good in the smear positive lepromatous patient, but its application may has its own limitations in the BTTT cases. If they are approved BT cases, why there is no 100% specificity? So if it detects only 25 to 60%, that's one of the limitations. It doesn't detect all the patients, it detects only at the most 60 patients. So we need to improve the sensitivity or the specificity of this particular technique. And one of the drawback of the PGL system that has been recent report also shows that it also cross-reacts with some other, for example, leishmaniasis. There's some degree of cross-reactivity is noted with leishmaniasis. In Indian situation, leishmaniasis is fairly common. So this is one of the fear is that it may not be very suitable. Anti-PGL as a seromarker may not be very useful in Indian situation. So we have to keep all this in mind. MLO4 or monoclonal antibody specifically reacts with 35 kD antigen of M. leprae. So that has also been worked upon. A fusion construction protein of two proteins, MLO405 and ML3233, these are all code names, don't worry about that, designated as LID1, showed diagnostic potential cases as well as household contacts. Now, I'm not saying that it is all ready to use, but these are the areas that are being explored and shown potential. Now, from serological markers can prove to be potential alternatives. The specificity of these antigens are advantageous in differential diagnosis of leprosy. ELISA and lateral flow assays are being developed on this basis of serological tests. Such tests can be implemented for routine use, but still needs to be worked on. It's not that final. So there are some identified tools which listed out by Madam. This is these are the main tools and markers that are in pipeline now that are being very intensively followed and by various laboratories in India and abroad. That is NDO lead complex test assessed using a smart reader. Now somebody has come up with a very brilliant idea. This is a spot test and the result is conveyed through a smartphone. So it is being transferred to the patient using a smartphone. This is called smart reader. Leprosy detect fast ELISA is also one of the quick tests. And UCP, LFA, interferon, that includes interferon gamma and or IP10 responses to ML2478 M. leprae antigen, specific M. leprae antigen. This is another test that is being tried by IL-1 beta, MCP1 and MIP1 beta. This is another test. And antigen PGL1, as I said, there are certain limitations, but it is supposed to have a good sensitivity in lepromatous leprosy patients. So if one wants to use in lepromatous patients instead of skin smear, if you want an alternative, this may be a good option. So real-time or the quanti-PCR based assays, as I said, like 16S RNA based assays can be used for detect the quantum of viable load in a bacteria in a given individual to say, okay, how effective or how efficient is the treatment? Is it bringing down the quantum of viable load to a acceptable limit? So that can be used as a monitoring device. So since they are not very expensive, so they can be utilized on. So these are the areas that are being that has a good potential, and it can be probably sooner or later will come into use in the real time situation. And RLEP real time. Conventional PCR using RLEP gene region is also another area that is being investigated. And for reaction neuritis, how do you differentiate? Is there a tool for predicting reaction? Do we have a tool for predicting reaction? Because that's most important. If you can predict reaction, you can stop reaction and you can stop nerve damage. So that is an important area as far as the leprosy is concerned. So people are trying to develop immunological, neurological markers. Immunological because reaction brings about all the cytokines